Hello everyone, welcome to today's webinar titled Development of a Blending Design Space Model to Control Uniformity in a Mini-Batch Direct Compression Line, hosted by BioPharma Webinars and presented by Manel Bautista, who is Senior PAT Expert Principal Scientist at Roche, followed by also joined by Bernhard Meir, Head of Continuous Manufacturing at Gericke, and lastly, Sebastian Sowinski, who is Key Account Manager at Sentronic. So, Manel, Bernhard, Sebastian, welcome. Thank you all for joining me. It's a pleasure to have you here. Again, a quick reminder to our audience before we begin. I'd just like to inform everybody that we will be having a live question and answer session after today's presentations before the end of the webinar. So please feel free to send in as many questions as you like via the questions tab, and you can find this directly below your webinar screen. And we look forward to going through your questions later. And please also include in your questions if they are for Bernhard or for Manel or for Sebastian. Okay, that's very helpful. Now, please allow me, of course, to introduce our first presenter. So Bernhard Meir is a research assistance, and during his diploma thesis at the Technical University of Munich, Bernhard studied the influence of continuous blending parameters and dampening of feed rate fluctuations. He joined Gericke in 2013 as a process engineer and gained extensive knowledge and expertise in powder handling and processing. Since 2017, he has been leading the Department for Continuous Manufacturing of oral solid dosage forms at Gericke. But with that done, I'd like to hand over to our first presenter today. So welcome, Bernhard.
Thank you, Marcus. So I hope you see my screen. I can start my talk about mini blending or continuous manufacturing of highly potent active ingredients in small batch sizes, because that's where the technology finds its most use for. First of all, just a very boring slide on the Gericke company. We are a Swiss-based company which was founded more than 125 years ago. We're a family-owned company in the fourth generation by Marcus Gericke, who you see on the right-hand side. And our focus has always been on powder handling and powder processing, specifically for loss and weight feeding, continuous and batch blending, but also pneumatic dense phase transport, which has
over the should be interpreted just like Spectra normally would. So if you have kids in an area that you
I just had
say carbon.
Quick difficulties. Yeah, and also pneumatic dense phase transport, which become quite a hot topic in the space of continuous manufacturing recently as well. We're roughly 350 people, and we worldwide presences all over the world to also support you on site if needed. So let's get into the topic of continuous manufacturing, specifically of oral solid dosage forms. When you talk about continuous manufacturing, you can obviously also talk about continuous manufacturing of drug substances, but my topic today will focus on drug product and specifically about the blending part of drug products. So first of all, I always like to look at traditional batch manufacturing and how that is done these days. So usually it starts with the manual dispensing and weighing of your ingredients, your active pharmaceutical ingredients, obviously, your excipients. Then they're introduced into a blender, which is usually a bin blender or a V-style blender. The powders are being blended, and then they are brought to the next transfer process steps, which can be dry granulation, wet granulation, or in the most simple case of direct compression, directly to the tablet press. And the philosophy of continuous manufacturing at Gericke is rather simple. So our process idea of continuous manufacturing is that we remove the IBC and replace it with a miniature continuous feeding and blending unit. We like to call that the Gericke formulation skid. And this formulation skid comprises of so-called gravimetric or loss and weight feeders that each meter a correct amount of powder into a small blender, which then continuously discharges into the next unit operation, which can be a tablet press, a roller compactor, but also extrusion or continuous wet granulation. So this here is a nice picture of a fully integrated continuous manufacturing line. So basically, material flow starts here at the bottom corner. Then we have transport to the first feeding and blending unit, where we can mix the intergranule phase. Then we can discharge into a continuous twin screw granulator, a truly continuous dryer. And then we can take the granules to the second feeding blending step, where we usually add the extra granule phases, the disintegrant, the lubricant, etc., and then discharge directly into a compression machine. And from there, we can take the tablets into a semi-continuous coater. And you see here, marked in red, is always the feeding blending unit at kind of the key of the process. This modular approach, the exact same feeding blending unit can be used for all different manufacturing routes. So if you want to do continuous direct compression, you have a single feeding and blending unit feeding directly into the tablet press. You can use the same setup also for dry granulation, where the exact same feeding blending unit feeds into a dry granulator, or the shown example where you have the continuous wet granulation process. Now let's look a bit more into detail of such a Gericke formulation skid. So usually these skids, they compromise, first of all, of the material handling, so the IBC discharge station, the containment handling, etc. And then we can integrate up to 6 loss and weight feeders. And in a truly continuous line, all these 6 loss and weight feeders, they feed a continuous mass flow of powder into the continuous blender, where we homogenize these powder streams. And then usually after the blender, you have some sort of PAT tool to assess inline the homogeneity of the blend and potentially reject out of space material, and then discharge into the tablet press or any other downstream equipment. We've been building these units now for several years, quite successfully as well. However, we saw some limitations in truly continuous blending. So first of all, if you want to dose a very, very low dose of API and very cohesive APIs, it's usually not possible to feed them very accurately at very low flow rates. So for instance, feeding a micronized API 50 grams per hour can be very, very challenging. And if you can't feed it accurately, obviously the whole process idea of continuous manufacturing doesn't work. Another challenge that we've seen is that these truly continuous processes, they have certain startup and shutdown losses. So it needs a certain time until the system reaches a steady state, or sometimes called the state of control. The same goes if you shut down the system, there will be residual powders in the system, which are basically waste. And these startup and shutdown losses can range to several kilos per batch. So each batch, you may end up wasting quite a lot of material. And if you're running a very, very low batch size, for instance, of just 50 kilos, and you lose 5 kilograms every batch, obviously this will not be very, very efficient. Also, in a truly continuous line, powders are continuously in motion. So this also makes the whole control strategy quite difficult, because, for instance, if you detect a fault, the powder has already traveled down the process line. So maybe you need quite advanced PAT sensors or RTD-based model predictive controls to compensate for that. And another, it's almost an oxymoron, is that the API consumption is relatively high. Although everyone got sold on the idea of using continuous manufacturing to reduce API consumption, which is still true for compared to the old batch process, you actually have to use quite a lot of API during the initial development of a process. And I'm sure you all know at the beginning of developing a new process, usually the API is very scarce and usually also very, very expensive. So these were kind of the 4 main drivers which led us to develop a different approach to make continuous manufacturing suitable for low dose and low volume. And that's what we call the mini blend process. So again, here is the schematic, and you see the material handling and the loss and weight feeder part is completely identical to a truly continuous line. However, the feeders operate in a slightly different mode. So the feeders are not trying to dose a constant mass flow now, but they rather do discontinuous feeding. So they want to have a certain mass that they want to feed accurately. And basically we feed this certain mass into a small mini blender, hence the name mini blending, which also operates in a discontinuous mode. So we feed a certain amount of powder into the blender, we close the inlet of the blender, we start the actual blending process to ensure a homogeneous blend. And after that's happened, we open the outlet flap of the blender and discharge into the tablet press. And with a small buffer volume above the tablet press, this semi-continuous approach becomes a continuous manufacturing line. So with the buffer, we can kind of run the tablet press without starting and stopping. And this is kind of perfectly aligned also with the ICHQ 13 guidelines, which state that continuous manufacturing can also be a combination of discontinuous and truly continuous process steps. But let's look into a bit more detail how this process works. So first of all, the feeding is still at the heart of the process, but it's discontinuous, and we're trying to dose a discrete weight, and this dosing of each of the individual feeders can either happen parallel or in sequence. And most importantly, since it's not a constant mass flow, we're trying to achieve things like feed factors, and establishing of feed factor profiles become much less important. And another big benefit is after you've kind of dosed this discrete amount of weight, the feeder is stopped and you can refill it. So refilling, which is usually one of the most critical process steps in loss and weight feeding, is done while the feeder is being stopped. So there's no risk of the refill actually impacting the feeder. And how the feeders operate, basically you start with a rather fast screw speed. So this is the so-called fast feeding, and I'll play a little video here. So it runs at a high speed, and at a certain switch point, it reduces the speed of the screw to achieve a high accuracy. And once the feeding has stopped, you can actually evaluate the mass that you've been feeding. So you can do a mass balance of all the individual feeders. So now, just to give you some examples, some data, where we try to feed 10 grams of micronized paracetamol. So this would be somewhat equivalent, if you say it's 1% in your formulation, to 100 grams per hour in a truly continuous line. And the numbers are relatively impressive. If you compare it to a truly continuous manufacturing line, we could achieve an accuracy of 0.16 with a relative standard deviation of 0.34%. So even these very, very low dose of a very bad flowing material, we can feed accurately in this discontinuous mode. So once all the feeders have completed the cycle, obviously the next step is the blending of the powders. So here on the right, you see a little picture of this mini blender, and it's basically a horizontal pedal style blender with a rough net volume about 10 liters. And these pedals, they rotate and divide the blend into smaller sub portions. With like its high speed and intensity, it can ensure a very high degree of homogeneity by convective and dispersive transport. Also, a benefit of these horizontal mixers is that they have ideal axial rail dispersion, and they limit segregation during the discharge. So if you compare it, for instance, to vertical blenders, these blenders are more efficient in blending low-dose cohesive material, and also they limit the segregation of the powders during the discharge. So again, here's a small video that kind of shows the blender in operation and kind of the turbulence it creates. And here on the right-hand side, we did 15 consecutive mini blends. And of each blend, we took 9 samples of roughly about a tablet weight. And you can see that the relative standard deviation inside the blender is way below 3%. So this shows you that even for 1% micronized paracetamol, we could achieve a very homogeneous blend. Then obviously the next step would be the discharge of the blender. And one of the key design features of this mini blender is that it has a very, very tight clearance between the housing and the pedals. So basically, when you're discharging the blender, you want to have as minimal residual powders inside the blender as possible. This is obviously down to like how much powder you can use to compress into the tablets, but also it's an important factor if you have to consider the carryover from one mini blend to the next mini blend. So it's part of the whole mass balance that you have to do. So next step, obviously, after the discharge is the tableting. And here you see some results of label claim of compressed tablets. On the left, you see a coarse API, so it was a coarse grade of paracetamol. And on the right-hand side, you see a fine grade, so micronized paracetamol. So what you can see in this slide is that if you have a very good flowing API, blending is achieved within very, very few seconds. However, if you have a micronized paracetamol or a sticky material, you will potentially need longer blending times to achieve this high degree of homogeneity. Another interesting thing we found with mini blend is now here we varied the excipient lot. So it was still micronized paracetamol, but we used spray-dried lactose, anhydrous, or granular lactose. And what's interesting to see here is that the pattern of label claim or content uniformity over time is very comparable. So independent of the excipient lot that you're using. And this is quite an interesting fact because this allows you now to vary the excipients in your blend without having to worry too much about label claim or content uniformity in your finished dosage form. So during formulation, you have a lot more degrees of freedom. However, so instead of doing just feeding, blending, discharge, we adjust the cycle slightly. So basically, we do a first feeding step where you would feed your API and your excipients. You would do a high shear blending step. So this would be at a relatively high RPM for a relatively long time. And then you would do a second feeding step where you would add the lubricant and you would blend at a much lower RPM, also a much lower time. And in addition to that, after each feeding step, we can do a mass balance. So we knew exactly how much API we fed, how much excipients we fed. And based on that, we can calculate the ratio and decide if kind of everything has met its target. And if not, we can do redosing of components. So it's a bit like you bake a cake and you realize you've put in too much salt, you put in additional flour to compensate for that. Obviously, this can't be done in an infinite way, and at a certain stage, we can also divert material from the process. And this diversion is usually based on soft sensors. So it's the mass of the feeders, it's the speed of the blender, and it's the time of the blending, for instance. And also, the diversion is happening directly in the blender. So since we're kind of diverting directly where the mistake happened, we also don't need to have any RTD models to kind of reject material, since it's really being rejected where the mistake has happened. We can do the same after the feeding and blending of the lubricant, and then eventually we discharge into the tablet press. where, of course, in the tablet press, you may need to consider again the residence time distribution from the little buffer hopper above the press, and you may have additional PAT sensors, for instance, in the feed frame of the tablet press. But I'm sure Sebastian and Manel will talk about this as well. So, short summary about my talk. The mini blend technology is suitable for low-dose micronized APIs. The feeding process is actually quite simple. So this also has the benefit that optimizing and setting up the feeders require much less time and eventually also API. We do not really have startup and shutdown losses because the first initial mini blend is exactly the same as the very last mini blend. So basically, startup and shutdown losses is whatever is left in the tablet press. With a net volume of 10 liters, we can produce mini batches between 2 and 8 liters. This also means that for initial development, you can actually use, like you see here on the left, a standalone mini blender, and you can produce a single mini blend, which is already of the same quality as if you would produce multiple blends. So it's a tool that you can use very, very early in the launch of a new product. And the combination of these also kind of results in less API consumption during development, and obviously then later also during tech transfer from development to your commercial line. The technology itself, it's suitable for rather low mass flows. So we targeted it as a mass flow rate from 1 to 20 kilograms, and there will be no scale up from development to the commercial line. I've given you a slight insight on control strategy, so it can be heavily based on soft sensors to assess critical process parameters, and PAT is seen more as an option. And also the material tracking, since everything is kind of being moved in discrete blocks, it becomes a lot easier because there's no or little need for RTD modeling also for the material tracking. So from the material handling all the way down to the tablet press. And same as our truly continuous lines, it's suitable for high containment, so it's up to OED5, and obviously with that also wash in place for safe dismantling after you have washed it down. So I hope I could give you like a short introduction to the technology, and if I've caught your attention, please feel free to reach out to us. We have a range of portable Gericke formulation skids available, which you can either rent for on-site trials, or you can come to one of our test centers worldwide to kind of experience this technology. So it can be either where you do tests on standalone feeders, standalone blenders, or fully integrated into a direct compression line to kind of get the full experience of mini batch blending. So thank you very much for your attention, and looking forward to your questions.
Okay, thank you very much for that, Bernhard. And if you have any questions for Bernhard, then you're welcome to send in as many questions as you like. We've had some already, which is very nice. Thank you for those. And we'll go through all of the questions later during our live Q&A session. Okay, now please allow me to introduce our next presenter, which is Dr. Manel Bautista, who received a PhD in analytical chemistry from the Autonomous University of Barcelona in 2009. Focused on the use of PAT in the pharmaceutical field. Manel joined Croda Chemicals after receiving the PhD, where he was implementing NIR spectroscopy as process control and monitoring of chemical reactions. Manel joined Novartis in Basel in 2015 as PAT expert, implementing real-time release testing for all solid dose forms, and since 2020, he leads the control strategy and PAT elements for the drug product continuous manufacturing line at Roche. And Manel has authored over 20 publications and given several presentations at international conferences. And beyond his current role, he is co-chair of two IQ working groups, the Drug Product Continuous Manufacturing and Uniformity Testing. And with that, I'd like to hand over to our next presenter. So welcome, Manel.
Many thanks, Marcus, for the introduction. Can you hear me?
Yep, I can hear you nice and clearly.
Okay.
Yep.
Okay, thanks. So good morning, good afternoon, everyone. So in the next minutes, I would like to present our proposal at Roche to develop and verify a blending design space model for our mini batch continuous direct compression line. I will go first through our mini batch CDC line, giving some highlights on this technology to proceed then with the work we have done through the blending design space model. So just for clarification, Bernhard just gave an excellent presentation. He was mentioning mini blend. When I'm calling it now mini batch, basically we are referring to the same, just for clarification. Good. So here you can see a graphical representation of our mini batch CDC line from to bottom. So we have the automated refill of high volume compounds using pneumatic transfer to the losing weight feeders. In our current setup, we have 4 losing weight feeders installed, but this line has capacity for 2 additional feeders. So with this, we can fit in parallel API and excipients, although we could also fit sequentially, as mentioned before. The material is fed into a plug shear mixer that we call it ourselves mini batch blender. And in our case, this is a 5-liter blender that has capacity to accommodate mini batches or mini blends between 0.6 and 2 kilograms, depending on the, of course, on the density of the material. So for our current work in the line, we are targeting a mini batch size of 1 kilogram. The fed material is blended in this mini batch blender, and after blending, the material is discharged from the blender, where we have a weight control and weight verification, where we can see whether there's material left in the blender. And you already saw that in some pictures from Bernhard before. So after this weight control, the material goes through a pipe, where we have the option to take samples as well as to measure with PET probes, kind of stratified sampling, while the mini batch moves through the pipe. After the pipe, we have one of the most relevant points on our line, which is the material diversion, where the mini batches with desired quality will progress, will advance to the tablet press, but mini batches would go to waste or to quarantine. So all mini batches are compressed sequentially, and the tablet cores are then transferred to a coater as a separate unit operation. Some benefits of using the mini batch CDC line compared to the standard or conventional continuous direct compression are no waste. So already, as mentioned before by Bernhard, the first mini batch is good material, as by working via mini batches, no calibration phase is needed compared to the standard continuous direct compression. Then only good mini batches, good material will reach the tablet press, making sure, of course, that the tablets that we are producing are of good quality. So this technology promotes as well using lean control strategies thanks to the excellent performance of the losing weight feeders. We've seen some examples before, as well as the mini batch blender. So no need for RTD, as mentioned before. And also important here is that this technology promotes the line ensures fully containment, allowing therefore handling of high potent APIs and also the vertical alignment for effective wetting in place cleaning. Another benefit of this technology is the absence of the technical transfer. So this slide shows the setup for the technical development, the process development in clinical supply, and finally, the commercial manufacturing one. So the technical setup consists in losing weight feeders, a mini batch blender, and a tablet press. So the 3 operations are disconnected, and this setup is used for formulation development, assessment of the disconnected unit operations, and to establish a blending design space model. The second setup consists an integrated line. So this is all unit operations are connected, so an end-to-end process development can be done. This line is intended to manufacture clinical supplies and primary stability batches, and we call it integrated line 1. And basically will be fully qualified next year. So finally, we are now working on the commercial line, and this will be exactly a copy of the integrated line 1 to be used in the commercial area. So as mentioned before, there is no transfer during development. And main reason to say that is that the fact that all configurations use the same losing weight feeders, so the same models, the same size of the losing weight feeders, the same mini batch blender with identical mini batch size, and also we use a conventional rotary press. So basically, this reduces dramatically the effort to bring a drug product from development to commercial facilities, as well as to give the opportunity to manufacture primary stability batches in the integrated line 1, which is sitting in the development facility. So how do we produce those mini batches? So this slide shows the mini batch genesis. So it all starts with the feeding of the API and excipients using the gravimetric mode of the losing weight feeders. After the material is fed in about 2 minutes, blending takes place for another 2 minutes. The material is emptied from the mini batch blender where the weight verification takes place, and then the mini batch goes, of course, if the quality is the desired one, to the tablet press. So in total, 6 minutes from feeding to its transfer to the tablet press. At this moment, the feeding of the second mini batch takes place, having therefore a sequential manufacturing of consecutive mini batches that will meet only the tablet press and having a line throughput approximately of 10 kilograms per hour when we have a mini batch size of 1 kilogram. As mentioned before, the material diversion point after weight verification is one of the most critical controls on our line. So we bring a mini batch to the tablet press or to waste based on 3 criteria. So the blend assay of a mini batch must be within a certain limit, typically between 95 and 105%. In this case, the blend assay for a mini batch is calculated basically with the losing weight feeders data. So we know exactly the amount of API and the amount of excipients that we feed into the mini batch blender. So we can basically easily calculate the blend assay. The second criteria is the blend uniformity of the mini batch. And the last criteria is the carryover or residual mass. So this is the material left in the blender that is calculated during the weight verification. When those 3 criteria are met, the mini batch will progress, will move to the tablet press. When any of those 3 criteria is not met, the mini batch would go to waste or to quarantine. 2 of those criteria are calculated based on feeders data and weight control. This is the blend assay and the carryover. The third one, the blending, will be formulation dependent, and I will show in the next slides our proposal to confirm the blend uniformity for all mini batches based on the blending design space model. So how do we generate the data to develop and verify a mini batch blending design space? So as mentioned before, we have exactly the same mini batch blender in our technical lab and the integrated lines for clinical supplies and for commercial. So therefore, we used a technical blender that gave us flexibility to handle easily our formulations. So here we have some pictures of the mini batch blender. On this one, here in the middle, one can see inside the blender with the metal blades. For all experiments that we did and that I will explain in a few seconds, so we discharge every mini batch into a vessel. This is the graphical representation of a vessel where we took for each experiment of the DOE, for each mini batch, we took 10 samples according to this representation here. And for all those 10 samples per mini batch, we then measured at line and Raman spectra. So which experiments did we carry out? So we performed a full factorial DOE, where we evaluated 4 factors: the mini batch size, between 0.6 and 1.4 kilograms. We had a target of 1 kilogram as a reminder. The blending speed between 230 and 280 RPM. The blending time between 30 seconds and 2 minutes. 2 minutes is actually our target as a maximum time for blending. And finally, the particle size distribution of the API used for these experiments, which is the same as shown before by Bernhard. We used as well APAP, acetaminophen, In our case, we used a 3% drag load, and the reason for us to evaluate a low drag load was to challenge the blending performance and to challenge how do we develop and verify design space model. So in total, 21 runs were performed, and the responses that were evaluated are listed here. So we evaluated blend uniformity by means of relative standard deviation of the Raman predictions. Remember, those 10 samples per mini batch were predicted by Raman, so we calculate relative standard deviation, and also the residual weight or carryover after each mini batch. Next two slides show the figures of merit of the Raman model that was done at Line at the time that we develop and verify this exact blending design space model. I'm not going to all details for this, which is, I believe, is not that relevant for today's webinar. Basically, we used at Line measurements at that time due to the fact that we didn't have by then the NIR probe that we have today after the blender. So therefore, we had to select an at Line measurement. It has rained a bit since we performed those experiments, and there might be today other possibilities on how to best use PAT to assess blend uniformity on individual mini batches, and we'll hear more about that from Sebastian in the next talk. But here the most important thing was to find the formulation blending design space. We wanted to have data from blending. So this slide shows the table with the results for each DOE trial for all the 21 mini batches, with the column with the assay values for each mini batch based on the mean value of the 10 Raman predictions, standard deviation of those predictions, as well as the relative standard deviation for each mini batch. Regarding the acceptance criteria of blend uniformity in terms of relative standard deviation, here we use the max value of 5%, which is typically a limit used when doing blend uniformity testing in batch mode. And from all those 21 experiments listed here in the table, there are 3 which are marked in red. Those experiments showed a high relative standard deviation above 5%, meaning that they showed high variability in the assay of the 10 samples compared to the other mini batches. So when looking at those conditions of the trials, we could already see that those mini batches used the lowest blending time and the lowest particle size. In this case was as well micronized APAP. So when doing a more extensive data analysis, we have here in the left side scatter plot of the blend uniformity response, where already looking at the color lines, we can see that there's significant impact from the API PSD, the particle size of the API, and also on the right-hand side here from the blending time. The impact for blending from the mini batch size and the blending speed is much less significant. So this also, when we look at the table, this also matches with the statistical data analysis from the DOE, where again, only API, PSD, and the blending time have a significant impact on blending performance. Interactions between the factors and quadratic terms were as well calculated, as in the table, but none of them was found to have a significant impact on the blending performance. So at this time, we had developed a blending design space, but if we think that we might want to use this in the future, in the commercial line, we had as well to verify the blending design space model. This is also in line to the ICH Q8, Q9, Q10 points to consider that describes the need of verifying a design space model. So we did that. We performed 6 additional trials. As we don't have yet the commercial line, but considering that we will have the same mini batch blender, we did all experiments in our technical mini batch blender as well. So we performed those 6 additional trials, which are listed here in this table. We kept the API PSD and the blending speed at target while we reduce the ranges for time and batch size. Reason for keeping the API PSD at target was that the model found the API revealed that the only lowest particle size, the micronized, was prompt to bring blend uniformity values above 5%. So we wanted to use the next level of API particle size that allowed us to have a good mini batch in respect to blend uniformity. From those 6 trials, purification mini batches, you can see on the right-hand side the relative standard deviation from the Raman predictions of those 6 mini batches, confirming what we expected, that we have good blend uniformity from those 6 purification DOE, where we expected, based on the conditions from those mini batches, based on the design space model, to have as well good blend uniformity. Looking now more in detail in the blending design space model, here we can see the contour plots of the BU prediction with the design space. So considering that blending time and API PSD were the most relevant factors for the blend uniformity, we show here 6 plots at 6 different conditions for the other 2 factors, the mini batch size and the blending speed. And we can see the impact in blend uniformity from the blending time in the x-axis and from the API PSD in the y-axis, and the response in blend uniformity is, as mentioned, in terms of relative standard deviation. We mentioned before that we set our limit on relative standard deviation for blend uniformity in 5%, but as we didn't want to put blend uniformity at risk for any mini batch, we proposed for this design space to have a tighter limit, in this case of 4%. So in the contour plots here, all the gray areas belong to conditions that would bring to a mini batch with a blend uniformity value and relative standard deviation values above 5%, while the white areas here are the conditions that would allow us to have mini batches based on the design space with a blend uniformity below 4%. We selected those green squares as an example where we could fix blend time and the API PSD to a certain value. The outcome of this exercise is given in this table on the left, where we define it based on the contour plots. We define the ranges for all evaluated factors that will ensure producing mini batches with acceptable blend uniformity. So those were the ranges that we identified based on the design space model. So how this translates into our control strategy? This is our proposal for commercial control strategy with the diversion. Again, this is diversion point number 1, which is our main control from the line. Is given by the blend assay from the loss in weight feeders, the data that we generate during feeding, as well as the blend uniformity. In this case, blend uniformity is ensured via CPP and CMA. So we would use basically, for if we had to produce this APAP formulation at 3%, we would use those acceptable ranges to make sure that blend uniformity is good for each mini batch. In this case, we added as well, at tablet core level, we added a PAT at line measurement as an IPC to control content uniformity. So regarding the model categorization, since we are developing and verifying a design space model, we already had interactions with some health authorities where we proposed and got positive feedback on the categorization of those models. So the blending design space model is classified as medium impact model, as it's not the solely indicator of uniformity and is used to establish the design space, and therefore the CPPs and the CMA ranges, the ranges that we shown before, in order to ensure proper blend uniformity for each mini batch that we produce under those conditions. The PATCU model as an IPC was classified as a high impact model. So as summary, we are proposing to develop and verify a design space for blending to allow producing sequential mini batches and to ensure for each mini batch a proper blend uniformity. So a blending design space model has to be developed and verified for each formulation, where all CPPs and all CMAs that we identify during process development and that have an influence on blend uniformity must be part of the design of experiments, must be part of the evaluation to define the design space. Blend uniformity will be tested by PAT or any other analytical technology until the design space model is verified. Once acceptable ranges are verified based on this design space, blend uniformity will be ensured and not tested for each mini batch. And another aspect, regulatory here, so the registration this year will basically include the design space model for blending and will include the acceptable ranges for blending. And yeah, before I close my talk here, would like to thank my colleagues. So as you can imagine, this is not a one man show, a one man work. So there's a lot of people from my company working on that, and I would like to thank these and other colleagues. And with that, I would like to thank you as well for your attention. I'm looking forward to the questions at the end of the sessions, and now looking forward to hearing my colleague Sebastian. Thanks.
Very good. Thank you very much for that, Manel. And if you have any questions for Manel or for Bernhard, then please continue to send in your questions via the questions tab found directly below your webinar screen. And we look forward to going through your questions again later. Okay, so up next is Sebastian Sowinski. And Sebastian is the key account manager in the spectroscopy business unit at Sentronic in Dresden. So in this role, he is responsible for customers in pharmaceutical industry and supporting them from the first proof of concept for PAT implementation to the final installation and qualification. He is also the main contact and support for internal partners at Sentronic. Sebastian holds an MBA and joined Sentronic in 2000, and starting in the spectroscopy business unit and providing PAT solutions to the pharmaceutical industry as early as 2004. He gained a comprehensive knowledge in near-infrared spectroscopy and multivariate data analysis, as well as in all aspects of the application of PAT in different stages of the development and processing of solid dose pharmaceuticals. Okay, so with that, over to you, Sebastian.
Thanks, Michael. Thanks for the introduction. Thanks to Manel and Bernhard for this interesting talks. Good morning, good afternoon, maybe good evening to everyone here. I'm pleased to have a short presentation as a third one today, and I'm going to focus a bit more about NIR-based PAT, not that much on soft sensors as we had before. Yeah, maybe we will follow up in the Q&A session on this. Allow me to have a short introduction to Sentronic and in particular to the process, to the business unit process spectroscopy. Sentronic was founded in 1993, so 1st of April this year we had our 30th anniversary. Our main office is located in Dresden in Germany, and early last year we opened our subsidiary in the US, which is Sentronic US Corporation, and has its office located in Norwalk in Connecticut. We have two business units within Sentronic. One is developing an optical oxygen sensor for monitoring of dissolved oxygen in water, which is dedicated to be used in industrial fish farming. The business unit I'm working in is providing NIR PAT solutions or NIR solutions. And within NIR, we have a very strong focus on diffuse reflectance. And this brings us to one main market, which is PAT for OSD, pharmaceutical processing. And we have a second product, which is a handheld NIR, which is used for chemical identification in German pharmacies. Within process spectroscopy, we do have the CentrPET family, which nowadays has 3 members. On the left bottom side, you can see the CentrPET FO in combination with the CentroProbe DLS. This system is quite flexible and can be used in many different unit operations, as for example, granulation, drying, blending, and compression. A few of these processes are unit operations I will talk about later. It is applied in continuous as well as batch processing, as blending is still done very, very often in a batch bin blending process. We do have the CentrPET BU and the CentrPET DA shown on the right bottom corner. And these two units are fully dedicated to the requirements of batch blending. The technologies we are using on the left top corner, you can see different spectrometer technologies used to generate a near-infrared spectrum. We are using the tunable laser system, which is based on a MEMS module in the central part BU, and all other systems we provide to our customers are based on dietary spectrometer technology. Just in short now, I will talk about a bit more in detail later. My talk today focus on monitoring a continuous direct compression process. And within such a CDC, we do have 3 different unit operations. Manel, as well as Bernhard, already talked about. So we have feeding, we have blending, and then in the end, we have compression. Each of these unit operations within the CDC line has its CQAs and CPPs, which can be assessed using NIR-based PET. NIR-PET nowadays is widely commercially adapted and used to assess material properties, post-blending, and pre-compression. Of course, there are also measurements on a tablet. The very last step we have our systems installed is in the feed frame of a tablet press. Short excursus on NIR-based PET systems in CM. Yeah, this slide is probably to majority of people not a new one. Why we use PET at all? Actually, there are 2 main drivers to use PET, not only NIR-PET, PET in general. In the first instance, these are regulatory requirements that PET is used to assess CQAs in real time. And this is done or might be required as part of its CM control strategy to be conform with, for example, ICH Q9 requirements. PET has, NIR-PET has a chemical selectivity. This is not possible to achieve with common soft sensor approaches. So a soft sensor might be used to show, yeah, process is under control, but PET, NIR-based PET or Raman do have a chemical selectivity to confirm this. The second key driver is, of course, the process understanding, and talking about the mini batch or mini blender is, for example, to evaluate the blend dynamics, the material properties during blending. Last but not least, from a process understanding point, that Manel had a slide in with the sampling, having PET installed inline allows a sampling density which cannot be achieved by any other sampling method. For example, with an assessment every 1 second, that would be at least challenging via a sampling approach. As based on the topics today, there might be a short reminder, very short introduction to NIR. This is a set of spectra shown, sorry, a set of typical NIR spectra, and these spectra contain physical as well as chemical information. Based on the raw spectra, it is possible to have a trending based on physical properties, but more often spectra are pre-treated by means of mathematical methods, and these pre-treated spectra can be used to extract the chemical information or also to trend the process as shown here. What are requirements for PET equipment? Not only NIR-based PET equipment, but of course, for Sentronic, the NIR-based is a more interesting one. So general requirements are listed on the left-hand side, and they are pretty obvious. So systems must be ready to be used under GMP. It must be possible to qualify and validate them. They must conform to regulatory guidances and to regulatory requirements. They have to be on the maximum possible technical level for long-term stability and reliability. A system owner always is interested in a minimum of service requirements, and they have to have multiple options for interface options to software and hardware. For CM installations, it partially adds that the systems have to be robust to variation in product velocity or material velocity. The systems have to be capable of running 24/7, and also an absolute minimize need of user or operator interactions. How do we address this, or how Sentronic is addressing this? It's with the CentroPad-FO and CentroProbe DRLS. The CentroPad-FO is a flexible analyzer platform based on a diode array spectrometer system. These diode array-based systems are very, very fast. The measurements are done within milliseconds for single spectra, averaging then typical measurement intervals are around 1 second, which gives the sampling density I just mentioned. This system is combined with different probe types for diffuse reflectance, as shown in the pictures. And the system is also able to be operated standalone, or it can be integrated into common process control software like CyPad, SyndiQ, PharmaNV, and others. What makes diode array-based systems fit for process use? First of all, they have no moving parts inside the spectrometer, so they are fully robust against vibrations, and they have a low risk of alignment over time, or if the system is subject to vibrations, or if the system is moved. Another very important feature for measuring moving materials, moving solids, is that all wavelengths are collected simultaneously at the same time, so there's no noise picked up if the sample presentation changes. Last but not least, it can be used with fiber optic cables. So there's a variety of probes which can be used and can be installed in the process. But of course, there's a however or but, diode array analyzers also present some challenges. First one is they are known for having a wavelength stability not as robust as FT or scanning systems. The intensity can drift over time. Last but not least, the resolution is not as high as for other technologies. For the last bullet point, resolution, NIR bands of many analytes in small molecule pharmaceuticals are broad enough that the resolution is still fit for purpose. How do we address the other 2 points? The first one is our wavelength standard, which is integrated into every system. And this is based on a Fabry-Perot filter, which creates a periodic constrictive destructive interference spectra. During factory startup and calibration, we calibrate every system against the NIST traceable standard, and afterwards we acquire one spectra of this internal wavelength filter, which is acquired at the time we know the calibration is good. And then during routine use, this filter is measured again, and in case there is any shift, this is corrected in order to bring it back to be physically correct. In a test in the climate chamber, we did a temperature profile between 15 degrees C ambient temperature to close to 30 degrees. And as you can see, the deviation of selected peaks of a WC920 standard has been very, very tight and well below requirements of EP or USP. The intensity drift is corrected based on an internal standard or active reference. So we do have the sample stream, we do have an internal standard, and by measuring them both, we can correct for any change in the intensity which is caused by system internal components, for example, a light source aging. The result of this is shown here. We do have one uncorrected absorbance, and we do see changes at one wavelength. The drift, overall drift, is light source reduction in intensity, but there are also other effects, like in the center, with a change due to ambient temperature. If we now look at the internal signal for the internal standard, we see it has pretty much the same characteristics. And if we consider both spectra or both intensity curves, we get the final corrected absorbance, which is close to 0 and well below 1 milliabsorbance unit. Going into a more practical level, more into the application itself, I have a picture here of a GEA line, which is used with many of our customers. And we have the direct compression part on the left-hand side with the feeding, with the mixing and the tablet press. And going back to the slide I've already shown, these unit operations do have their CQAs, which can be assessed using NIR PET. In feeding, that's material identification to ensure the correct materials are feeded. We have the blending where we can monitor the blend uniformity and potency. And finally, in compression, the blend uniformity and assay. NIR PET is widely used and approved by authorities. A bit of history of PET used here. So our first project we had within a CDC line was as early as 2010. Back at this time, the NIR was installed at the transfer chute between the mixer, blender, and the tablet press. It was combined with the diverter, and the application and measurement here was possible due to the features I mentioned about the dial array being very fast, not collecting noise, not picking up noise due to changes in the sample presentation. One key disadvantage of this approach is that the estimation of the sample volume is very difficult to not possible. And as a result, later on, the PET implementation was moved down into the tablet press, into the feed frame of the tablet press. Nowadays, this measurement is still done between blender and tablet press, but it is combined with sampling devices such as a SpiderView from Expo or the Gericke. Just a few slides on the feed frame of a tablet press. Here is a good example showing the central probe DRLS above modified pedals. There is a notch in the pedals, and this ensures that we continuously see material, or the NIR does see continuously material, and this measurement is done on or is possible on a 1 second interval, which provides a measurement on a relative large volume over a short time. So the time to unit dose equivalent is relatively short. I do not want to go into too many details about the feed frame, as it's not the topic of today, but just as a last slide on this measurement is implemented in multiple lines and approved for commercial manufacturing by regulatory agencies. There are unit operation simulators commercially available from Expo Process Analytics, also as shown here. And there are many, many, many publications. I've just listed 3 of them on this slide. Going into, or more into the topic of today's webinar, which is mini batch blending or the semi-continuous blending within a CDC line. We have, again, the 3 unit operations already shown by Bernhard and Manel. The semi-continuous unit operation here is a blending, as we saw, and a measurement in the chute, as just shown, is not meaningful anymore. There's a relative high amount of material which is passing by during the discharge of the blender, and in the time between, there is no material measured at all. The measurement in the feed frame remains a good position for PET, but it is also possible to move the blender as a PET into the blender and to really measure the blending and to monitor each discrete mini batch in real time and inline. We have been approached by Martin Bormann and Gericke a couple of months ago, and the discussion we started was, how can we measure in a GPM? First ideas we had was to measure from the side based on a probe with a curved window, but the disadvantage early identified was that the pedals will have an impact on the spectra. It is not possible to have notches in the pedals as we have for the feed frame installations, as this would impact the blend performance. And the option to detect and reject spectra during the measurement was considered as not being ideal. So in the next step, we started to think about how can we install a probe looking from the front plate, as shown here. And as a result, it was discussed to have a modified front plate to the Gericke system, to the Gericke mixer, with 3 positions between 6 o'clock and 9 o'clock, as we have not been sure on what's the best position, how will the powder behave or the material behave, especially with a lower load. And some pre-tests were done, and they showed that 6 o'clock position is the best suited position, in particular for low loading levels. This resulted in installation for some tests. And based on this setup, based on this installation, we acquired spectra on one batch, mini batch. I know Bernhard don't like that I call it batch. I do not know a better name yet. And this is a run of 1 kilogram with 10% paracetamol in. We also measured the individual ingredients. Here we had only lactose and paracetamol. We see the spectra. And looking into the pre-treated spectra of the entire run, we see 2 ranges with clear changes. And if we compare them to the raw spectra of the ingredients, we can see these changes are related to the paracetamol. Looking into some very simple approaches in the next step, we trended the wavelength over time, and we can clearly see how fast, how quickly the mixing is done in this process. Another simple approach to assess the blend characteristics here is doing a PCA, and we also see, similar to the wavelengths as plotted, trend in the first principal component. So this very simple experiment, very quickly done, has shown that it is possible to measure the blending, to monitor the blending based on a measurement installed inside the blender. The conclusions of this work done here is that dietary-based NIR systems are capable to measure extremely fast, and they can monitor also this very fast processes in a mini blender. In routine operation, the homogeneity can be determined inside the blender, and this signal can be used also to discharge the mini blender, which can be used to control this semi-continuous operation. And this measurement can also be part of a strategy for a real-time release testing approach in a CDC line. In the very end of my talk, just 3 slides about what's going to happen next. The first one is a new system we are working on, which is Central Path Compact, which is currently in development, and the launch will be next year. This will be based on a dietary spectrometer as well, and it will combine the spectrometer for the NIR range with all the features we have for the Central DLS. It will be configured with a controller based on Linux, with a power over Ethernet functionality, and based on an ATEX certification. It can be used for any powder and granule processes. The spectrometer we are going to use is the one we have already installed in our Sentronic A, and just last week I got some results about a test base where magnesium stearate in MCC was calibrated, and as you can see here, even very low concentrations can be clearly separated, and it is possible to build models. For the 1%, we even did the blending, and this was done in small vials inside here. The last one I would like to mention is a Save the Date announcement. Next year we will host the Sentronic AT workshop here in Dresden again. It will be in the first week of December, and it will be continuation of our PT workshop we did this year in the US, as well as the workshops we did prior to travel limitations. With this, thank you for your attention. Thank you for your participation. I'm open for any questions, comments, remarks.
Okay, thank you very much for that, Sebastian. And so now it's time for us to begin our live Q&A session. Thank you to everybody who sent in questions so far. You can still send in your questions if you have any more for Sebastian or for Manel or for Bernhard via the questions tab directly below your webinar screen. Please continue to mention in your questions which presenters they are for, because that's very helpful. Okay. Now our first question today, we'll go to Bernhard. Okay, so this question here says, regarding the last slide prior to the summary, what is percentage lubrication you usually go for, and how do you go about detecting it for validating the blending process has been performed accurately?
So with regards of lubrication, you will need similar amounts than in a batch process. And validation of the correct amount, it's also done in the same way. So basically you have to press tablets and measure hardness and see how that is impacted. Maybe Manel can share also some insights on that.
Yeah, I would say no, there's not much from my side. Basically, I would just repeat what you just said. So, yeah.
Yeah. Okay. And anything more to add from Sebastian before we move on?
Nope.
Nope. Okay, then. Thank you very much for that answer, then. And our next question will go to Manel. And this question here, Manel says, could you please elaborate more on the fact that blending speed was found not relevant for the development of a blending design space?
Yes. So, if I can remind, so in our design space, we set this factor, the blending speed, within the range between 230 RPMs and 280 RPMs. We found that blending speed was not relevant or had not significant impact on blend uniformity, but only at that range that we evaluated. We know, of course, that if we had blended at a very low speed, we would have an impact on blend uniformity. But in this case, since we wanted to make sure that we have a certain regime of blending, we used this range. And within this range, we didn't see any impact on blend uniformity.
Okay, that's great. Thank you for that answer. Any additional comments from Bernhard?
Yeah, so we saw something similar. So you need a certain RPM or you need a certain shear to disperse particles. But once you've exceeded that, it's more or less down to blade passes. So it's about the function of time and how many rotations you have. But you need a certain minimum RPM for high shear blending.
Thank you, Bernhard. And anything else to add from Sebastian?
No, all these are process questions, so I'm not the guy here.
Yeah, okay then. Thank you anyway. So this next question then for Sebastian says, why can't I just use soft sensors to assess and control a mini batch rig? Why do I have to deal with all this NIR PAT stuff?
Well, my hope was that I answered this already with my presentation. Yeah, actually, as mentioned, PET, NIR-based PET, or in general, any measurement which really is based on product properties, material properties, give you some chemical insight into the process. All this first principle models, soft sensors, all this stuff is good for use, but PET, again, not only NIR, is an excellent tool also to verify assumptions which are made to build first principle models, which are, or PET can be used to verify the soft sensor provides the correct information. And all this then is based on really chemical properties or sometimes physical properties of the material inside the system.
Okay, thank you, Sebastian. Anything more to add from Manel?
Yeah, maybe I would like to add as well. So I fully agree with Sebastian. I believe that, and basically from my experience, I've been working in the PET field for a long time, and I know that using PET during process development and formulation development is extremely useful. So I encourage everyone to make sure PET is part of the process development and formulation development. And in this case, yeah, from the example that we've shown here about the blending in a mini batch blender. So basically what we have seen is that PAT can support a lot, and is basically what we used to develop and verify the space model. But then we can mimic more or less what we have on batch process. But again, I mean, PAT is part of what we have shown today here.
Yeah, I agree with this as well. And I think one of the huge benefits of PAT tools, or most PAT tools, is the high sample frequency. So maybe the results I showed, we took 9 samples from inside the blender with a sample thief, which also you can question how representative it is and how much segregation you have in the sample thief. Whereas with the solution Sebastian is showing, with the NR integrated into Blender, we get several samples in a second. So also you get a real insight on what is happening on a very, very short interval of time, which is very, very nice and very powerful for process development.
The sample sieve is a good point, Bernhard. In the beginning, early phase of my presentation, I was thinking about, yeah, well, if I measure after the blending, this is something like a post-process analysis, and I was going to think about adding a picture of a sample sieve, because that actually would happen then.
Yeah, and we've actually seen quite a lot of cases where our customers would take blend samples, and they would realize these samples are completely off, whereas the finished dosage form is perfectly homogeneous, content is perfect. So it shows that physically taking a powder sample from the process has its challenges itself.
Yes.
Yeah. Also the blending happened in the feed frame then.
Here too.
Okay, I'll move on to our next question, and thank you for those answers. Our next question to Bernhard says, often there is need to mill micronized APIs. Is this also possible to include, or have you seen that there is less of a need for an immediate milling step because of the higher shear blending?
Mm-hmm. So in our truly continuous rigs with our linear continuous blender, we quite frequently integrate cone mills upstream the blender, because in a truly continuous blender, you have very, very limited shear that acts on the powder. However, in mini blend, the shear tends to be enough to disagglomerate as well and to ensure dispersion of particles. So there we would not integrate a cone mill. It's possible, but it poses some risks, because a cone mill by itself, you have chances of material buildup. That material could drop down in an uncontrolled manner into the blender, and potentially also risk of over or under concentration inside the blender. So we would not recommend that to you.
Mm-hmm. Thank you, Bernhard. And anything more to add from Manel?
No, not from my side, no.
Okay, and anything else from Sebastian?
Nope.
Okay, thank you then. And our next question to Manel says, would it be possible to use this design space if a higher throughput was needed?
Yeah, that's a good question. So my first answer would be, first of all, we would need to see the difference in the throughput. And then since we have a model, the idea would be to predict the blend uniformity based on the new throughput, which translates into the new mini batch size, I believe. And then most likely we would need to verify this design space model based on the new mini batch size that would allow us to reach the throughput that we desired. I would say that would be a way to do that. Another factor that could be increased or decreased at this point would be the blending time. So we would be able, if we would be able to blend in less time, we could as well try to increase the throughput. The example would be applicable to the blending time. We would need to verify whether the model is able to catch this variability on the blending time.
Okay, thank you, Manel. And anything more to add from Bernhard?
So if you're looking to increase throughput, obviously my recommendation would buy a second line from us and keep things constant, because I think that's the beauty about this technology, is that you don't scale up or you don't scale down, and you scale by numbers of lines.
Yeah, that was the option that I left. Yeah, true.
Very nice. And anything more to add from Sebastian?
Nope.
Okay, then. Thank you, then. So this next question for Sebastian, it says, when will the Centro Compact be available?
Yeah, actually, we plan and work towards to present it at IFPAC, which will be in Washington, D.C., first week of March next year.
Nice. Okay. And anything more to add from Manel or Bernhard?
I can't really.
Yeah. Anything else to add from Bernhard? No.
Okay. I'm happy. I'm looking forward to receiving one to get my hands on.
You will get one.
Very nice. Okay, our next question then to Bernhard says, well, it's a question to Bernhard and Manel. So let's ask it to Bernhard first. It says, what is the maximum throughput possible?
So basically, throughput is a function of mini blend size and cycle time. And now it depends a bit on the size of the blender, but if you assume you have a mini blend size about 1 to 2 kilograms, and usually can do about 10 cycles per hour, it's in the range of 20 kilograms per hour maximum. And if you want to go for a really low throughput, you just wait before you discharge the blender.
Okay.
So there's no lower limit like on a truly continuous line.
Yeah. Okay. And also, Manel, what's the maximum throughput possible?
Yeah, I mean, it was already mentioned by Bernhard in our case. So as shared in my presentation, our mini batch blender has a capacity of 5 liters volume, and this basically with powder, this translates more or less when we want to meet this 50% capacity of the blender. This translates in 1 kilogram more or less. And as mentioned by Bernhard, 6 minutes each cycle is more or less what we target. So this would end up with 10 kilograms per hour. Here it depends on the volumes that you have on the mini batch blender, and yeah, so there are several options, right, Bernhard? So
okay, thank you for those answers. And anything more to add from Sebastian? No. Okay, then. I'll move on to our next question for Sebastian, which says, just lost it. Hold on. There we go. Are there any sorts of OSD products that are incompatible with NAIR-PAT?
Actually, there are not many, especially in OSD. I saw once an API which was a bit boring with respect to the spectral properties, but usually I would bring it down to the concentration level. So if, for example, cancer drugs or something like this with concentrations, active concentrations of less than 1% API, I would consider them as least challenging to not possible. That's then just a monitoring of the overall process about excipients behavior and all this stuff. But to get chemical information based on the API in such cases, not very likely anymore.
Okay, thank you then. And anything more to add from Manel?
No, not from my side. Fully agree with what Sebastian mentioned.
Yeah, okay. And anything else from Bernhard?
I'll leave it to the experts.
Yeah. Okay, then. Our next question then to Bernhard says, what's the contaminant level?
So I guess it's containment level question, yeah.
So yeah, containment level, yeah.
Containment level, yeah. So the containment level we can achieve with like a standard, almost off-the-shelf mini batch blender or mini. Oh, I'm saying mini batch now as well. A mini blender is about is OEB4. However, all of our feeders and the blenders are designed to be integrated into an isolator. So all of them are kind of through the wall design. So the feeders and the blender can be put into an isolator, and then you can achieve same containment level as you would do with an isolator. So even up to OEB6 if you want to. However, where we limit it is the arrangement of the feeders in the isolator. You can't arrange 6 loss in weight feeders in an isolator. So you're limited by the numbers of feeders that you put into that isolator for the very highly potent compounds that you want to process.
Okay, and thank you, Bernhard. Anything more to add from Manel?
Yep. Well, maybe from my side, so with our experience having feeders in mini blender integrated, it's about a CAT 3B. At least this was our internal requirement, and this is what we achieved. And yeah, this is more or less what we can share from our side.
Yeah. Okay. And anything else to add from Sebastian? No.
Nope.
Okay. Okay then. Thank you then. Our next question to Manel says, what assumptions are you making to generate your BU contour plots? Do you see any risk that changes may occur during manufacturing that could violate those assumptions?
Yeah, well, basically the contour plots are based on the design space that we do from the statistical data analysis after the DOE. So what basically we did here is to come up with a design of 6 purification runs. So what we did basically to make sure that the model had a good predictive capability, we predicted those 6 mini batches, and then we compare with experimental results for those 6 mini batches. We saw that the prediction versus the experimental results matched within the predictive interval. And then for the control plots, what we used then was the higher values for each prediction, meaning if we had a prediction of 2% plus minus, let's say 0.5%, we took the 2.5 because that means that's the worst case on blend uniformity. That's how we set and the counterplots on the worst case. And to the second question was about, sorry, forgot.
So do you see any risks that changes may occur during manufacturing?
Yes, well, basically, so we are defining a design space based on a risk assessment, meaning we have identified CPPs, blending time, blending speed. We have identified CMAs, let's say API of the or a particle size of the API. If during commercial manufacturing we identify that there is a novel CMA that we didn't find during development, this of course this should imply a redesign and verification of the design space model. However, I mean this is not that really different compared to what we are doing nowadays, and since a lot of years in batch manufacturing, where we identify our CPPs, our CMAs. We do traditional batch blending, and this is not that different to what we are doing at the end, right, Bernhard? I don't know what you think.
Yeah, thank you.
Yeah, I think this is one of the great benefits also of this technology, that it has so many similarities to a traditional batch process. So yes. Also, what we've learned that a lot of companies, it's much easier also internally when they're talking to their quality guys, that they can always make this analogy to a traditional batch process, and it makes them much easier to understand things.
Thank you, Bernhard. And anything else to add from Sebastian?
Just a comment, Bernhard, you just mixed up batch and continuous again.
Yeah. Maybe why we're not calling it batch is because we had the feedback from regulatory that they found it very confusing if you use the terminology batch in a continuous process.
That's true. That's true.
So we try to avoid it.
Yeah. We try to make it more really sound and that everyone can listen about mini batch and. For instance, today, giving the webinar. But yeah, I understand and I see the point. Yeah.
Okay, very nice then. Thank you for those answers. And our next question to Sebastian says here, how many times does this LC internal reference occur? From my experience, we do struggle a lot with day-to-day variability when using NIR. How can you overcome this issue?
Not sure what with LC is meant, but with the internal reference thing, I have an idea. So it's about the drift correction and wavelength correction. So the internal active reference, and this depends strongly on the ambient conditions. So if they are very stable, this interval can't be longer. If they are let's say, more challenging. For example, having a probe installed close to a fluid bed system where you have a fast temperature changes, it's done in a shorter interval. In general, it's something between 30 minutes and 120 minutes that the system does this internal one. Not sure where this question is coming from, but day-to-day variation might be also the process equipment might be different, lots of the ingredients. So I would not always put the NIR or the PET on a question. It can be. And to be honest, I also saw systems which were not really as stable as they should be. But again, I would not only put the PET on the question here, and would look into more details what's in. But again, this active reference or internal correction is between 30 and 120 minutes interval.
Okay, thank you, Sebastian. Anything more to add from Manel?
Nope.
Okay, anything more to add from Bernhard?
Nope.
Okay, thank you very much for the answer then, Sebastian. And this next question to Bernhard says, what was the API content in percentage in your example plots of blend time versus homogeneity?
It was 1% in the blend. And the total mini blend size was 1 kilo, so we dosed 10 grams of API.
Okay, thank you, Bernhard. Anything more to add from Manel?
Nope.
No. Okay, nothing from Sebastian. Okay, thank you then. And our next question to Manel says, in your verification DOE table, the data suggests that as the batch size increases, the RSD decreases. That suggests the accuracy of the feeding is the performance limiting factor. Is there a possibility to improve the RSD performance by dispensing pre-blended materials?
Yeah, I mean, yes. So we saw that there's a slight impact on the depending on the mini batch size, which means depending on the amount of material that we feed. That's true. And yeah, that could be an option, for instance, to have a pre-blend. But of course, what we are trying to avoid here is to use any additional or pre-additional unit operation before we start. So basically, we would like to come up with formulation and a process development that allow us to directly have high accuracy and high performance of blending, defining a design space for each blending. Depending on the formulation for without the need of a pre-blend. But yes, that would be an option. And basically, just to close my answer here, we already evaluated that internally. We decided basically to try to avoid that. But yeah.
Yep. Okay. Thank you, Manel. And anything more to add from Sebastian?
Nope.
No. Anything else from Bernhard?
No, it's basically there will be a lower limit which the feeder can feed accurately, but you have other measures to play with, like the mini blend size, which you can increase to also increase the amount the feeder has to dispense.
Okay, thank you then for those answers. And this next question for Sebastian says, how do you avoid the powder/particles accumulated on the sensor and cause the measurement error?
It depends. In an ideal case, and pretty often there is no issue due to this. If you, for example, in the feed frame of a tablet press, and also I did not see it in live for the mini blender, as long as the granules and the powders introduce forces to the probe window, there's a self-cleaning effect. There are other installations, as I mentioned, the measurement in a chute, which can be combined, for example, with a spider wheel. There's also an option to combine this with a probe installation, which contains a wiper cleaning the window when it is necessary. So based on the acquisition of the entire spectra, it can be clearly identified. Window falling happens, there is no exchange of material anymore. So as said, it depends. And ideally, we do not need it due to the installation of the probe, for example, in a feed frame or in the front plate of the mini blender.
Okay, thank you, Sebastian. Anything more to add from Manel?
Not from my side.
Okay. Anything else from Bernhard?
No.
Okay. Thank you. This next question for Bernhard says, have you seen any impact of blender fill level on the final blend properties within the 2 to 8 liter range you mentioned in your presentation?
So we did several trials on that, and we saw within this range, you see actually very, very little variation in blend uniformity. And I think also Manel showed a slide where you see that there isn't much impact. However, you need to always have a certain headspace inside the blender to allow the powders to move. So potentially can go higher than 8, but we would not recommend, because otherwise powders wouldn't move and they wouldn't blend very well. And on the lower end, it's more to do with the residual amounts or the carryover in between mini blends, which becomes, well, as a percentage, significantly higher than if you run at 1 or 2 kilos.
Okay, thank you, Bernhard. Anything else to add from Manel?
Nothing to add. Bernhard explained it perfectly.
Yeah, okay. Nothing else from Sebastian?
Oh, wrong this time. Yeah, just at the NIR probe to see the blend curve in dependency of the filling level. So you will see a value every second.
Yeah, that's actually one thing we also saw with the initial trials. If you have too little powders in there, you don't get good sample presentation on the NIR probe.
Yep, not unexpected.
Yeah.
Okay, very good. Thank you. And this next question for Manel says, is the weight gain hopper in your system on a load cell to confirm dispense weights?
Yes, that's the case. Yeah, we have a load cell below.
Okay, yeah, very good. Very nice succinct answer. Thank you, Manel. Anything more to add from Bernhard or Sebastian? No? Okay, very good. This next question to Sebastian says, is there direct contact with the powder or measured through the window?
Actually, our probes, especially the central probe DRLS, is designed to measure in direct contact with the product. It's built from sapphire and stainless steel, so it's okay from a GMP point of view. It is possible to measure through a window as well, but this introduces some additional risks, challenges, and yeah, maybe we get these questions later on, so I can follow up offline, as this would really be too much for now.
Sure. Yeah, I get it. Thank you, Sebastian. Anything else to add from Manel or Bernhard?
No. Maybe just that you need to also consider the containment aspect. So if you want to avoid as many gaskets and sealing as possible, then a sapphire window would be preferable, but obviously not ideally from a measurement point of view.
Yeah. Okay, thank you then. This next question I think it's for either one of you three, so I'll just ask it to Bernhard first. It says, for the development of the design space model, did you rely on only PAT data, or did you do reference testing? If PAT, qualitative or quantitatively?
I guess the design space model was developed by Manel, but I would say both is possible. You can do PAT or inline sampling.
Yeah, that's correct. And in our case, we use PAT data only and quantitative, basically, to make sure we could predict and we could find the relative standard deviation for each mini batch.
Very nice. Thank you for those answers. Anything more to add there from Sebastian?
No. I guess it was a question to Manel, and he answered it, as I would do.
Very nice. Thank you. Then. This next question is to Manel and Bernhard. I'll ask it to Manel first. It says, have you seen a change in fill depth requirement at the press with longer blend times due to decentrification?
We haven't seen any impact to densification in any of those trials that we have done, not only from those that we share today in the DOE, but also from additional trials. We haven't seen anything. I don't know, maybe Bernhard, do you?
Not really, but I would assume, since you can operate the blender at relatively high shear, and let's assume you have a granulated lactose as your excipient, I think if you have very, very long blending times, you could kind of break the granules, and that would change the fill depth. But that's an assumption. It's not something we've observed so far.
Yeah. So I mean, we've done trials with some of our Roche molecules here, and only when we had extreme blending speed for a long time, it was more kind of a robustness test, and we might had perhaps some changes on the properties of the material, but it was very rare. No, I think with the regime that we are using, at least we haven't seen much on that.
Okay, thank you, Manel and Bernhard. Anything more to add there from Sebastian?
No.
Okay, thank you then. This next question for Bernhard says, you mentioned a lot of benefits of mini blend over truly continuous blending, but do you also see any drawbacks?
Well, there are a lot of benefits, that's for sure. However, there are some drawbacks, and that's also the reason why we kind of offer both mini blend and truly continuous, and that's the mentioned throughput. So we kind of said a rough figure is somewhere between 10 and 20 kilograms. But imagine you have a high volume blockbuster drug or generics manufacturing. You want to produce high volumes of products. I said one option is to buy more lines, which is great for us. But at a certain overall batch size or annual sales volume, the benefits of a truly continuous line with a higher throughput exceeds the one of multiple mini blend lines.
Okay, thank you for that, Bernhard. Anything more to add there from Manel or Sebastian?
Yeah, so maybe Bernhard already answered the question, and I think we are not anymore in the age of having really blockbusters. So I think we are going more to target therapies which require less volumes, and this is where I personally believe that using the semi-continuous direct compression will help those companies to adapt the manufacturing according to the demand.
Yeah, I guess at the end it depends on the portfolio you have as a company.
Exactly. Yeah.
Okay. Anything else to add there from Sebastian?
No.
Okay, thank you. So our last question for today then, it goes to Bernhard once again, and it says, how does the investment cost of a mini blend line compare to a continuous line?
So investment cost of a fully integrated CDC line is actually very comparable, because maybe you saw in the flow sheet that it shares a lot of common elements of a truly continuous line with a mini blend line. But I think the real cost-saving benefit of this technology is during the development of a new drug, since you use much less API. And I think Manel also showed an example that you can use individual unit operations like the blender in standalone mode. So it's obviously from a CapEx point of view much cheaper, but also developing your design space of the blender is easier because you can bypass the feeders, for instance. So that's where the real cost benefit of this technology is.
Okay, very nice. And anything more to add from Manel?
No, basically what Bernhard mentioned. Also, I would say this also has an impact. I would say long term. It's about implementing, let's say, more simple control strategies. I think it saves time during development as well, and at the end you are saving costs.
Thank you. And lastly,
with increased quality.
With increased quality. Very nice. And anything more to add from Sebastian?
Nope.
No. Okay. Well, that's it then. That was our last question for today. So thank you, everybody who sent in your questions for today's Q&A session. It's been really great to discuss them here with our presenters. And I'd just like to remind you that you can watch this webinar and other webinars by going to our website, which is biopharmawebinars.com/webinars. And if you're watching this webinar on demand and you have any more questions that you would like to be answered by presenters, then you're welcome to send your questions over to me. You can reach me on LinkedIn under Marcus Osei, Osei, and any questions I receive, I will very quickly email Sebastian, Bernhard, or Manel to get those answers back to you. Okay. But before I finish today's webinar, I'd just like to ask our presenters if they have any closing remarks. We can go in order of presentations. So, Bernhard, over to you.
No, I think it was a great discussion also after the webinar with a lot of good questions. I think this shows that there's a lot of attention to the technology as well, and I'm looking forward to staying in contact with you guys.
Very nice, thank you. And over to you, Manel.
Same for me. So thanks for all the questions. I think we had very, very nice talk today. So we covered 3 presentations. Really nice. So yeah, thanks.
Thank you. And over to you, Sebastian.
Nothing in particular to add, just thanks to Bernhard, to Manel, to you for organizing all this stuff, and thanks for everyone who somewhere joined us in the background. So we do not see how many people are online. Might be better in both directions. So thanks again.
Yeah. And I'd also like to take this opportunity to thank our presenters for joining us today. So once again, that was Manel Bautista, Senior PAT Expert, Principal Scientist at Roche. So thank you, Manel, for joining me. Also, Sebastian Sowinski, Key Account Manager at Sentronic. Thank you, Sebastian, for joining me. And last but not least, Bernhard Meir, Head of Continuous Manufacturing at Gericke. Thank you, Bernhard, and all of you for joining me in today's webinar. It's great to have you here. Some great discussions, and hopefully we can work together again soon. Okay, so I'll end it here. You're welcome to leave, and I'll speak to you very soon. Okay, take care.
Thank you very much.
Thank you very much. Bye.
Bye.